Hello?
Hi, Jack.
Yes. Hi.
So actually, the webinar will start at 10 a.m. So
Yes.
yeah, we still have 15 minutes.
Yes, I understand. But I just want to make sure I tested the equipment, make sure everything
Uh,
is okay. Yeah.
yeah, can you try to share your screen?
Okay, sure. Let's see. Are you able to see the screen?
Yes, yes.
Okay, my screen. Yeah. Okay. Let's see. Put it into the full screen mode. You can see it, right? I hope.
Yeah, I can say.
Right. Okay. Okay, it's good.
All right, yeah, it's good.
Hi, Susan.
Hi, Jack.
Good to see you.
Nice to see you. I'm looking forward to this.
Yeah, thank you for coming.
No worries.
I can see you in your office, right?
Yeah, I don't make it in very often. We're having a lunch faculty meeting today, a
Uh-huh.
little celebration for getting our breast spore sent out to the
Oh,
world to be reviewed. So,
that's
uh,
great. That's great. It's harder and harder to get
oh my
sport.
God, it's just
Yeah.
very challenging.
Yeah, but just need to keep trying, right?
Yep, I'm going to mute and let you do your thing.
Okay.
All right, so yeah, it's almost 10 a.m. Houston time, and I see there are already around 90 uh participants join this webinar. Maybe we can get started. So I can use the 3 minutes to introduce Dr. Jack Lee, and so he can have more time uh, in the webinar. So, good morning, everyone. Welcome to today's ISBA's webinar. And on behalf of International Society for Biopharmaceutical Statistics, it's my great pleasure to introduce my colleague and today's speaker, Dr. J. Jack Lee, for his presentation about Bayesian methodology in clinical trials. And the recent FDA draft guidance represents an important milestone for the broader adoption and appropriate implementation of Bayesian methods in clinical trial design and analysis. For many of us working in Bayesian clinical trials, this guidance is both timely and highly relevant, and it provides an opportunity to reflect on where the field has been, how Bayesian methods are currently being used in regulatory settings, and what challenges and opportunities lie ahead. And we are very fortunate to have Dr. Jack Lee share his perspective with us today. And Dr. Lee is professor of biostatistics and the Kennedy Foundation Chair in Cancer Research at the University of Texas MD Anderson Cancer Center. He is internationally recognized for his contributions to Bayesian adaptive designs, statistical computation and graphs, drug combination studies, biomarker identification and validation, and statistical methods for precision medicine. Over more than 30 years at MD Anderson, Dr. Lee has collaborated with numerous multidisciplinary research teams and has made major contributions to the design and analysis of clinical, translational, and basic science studies. He is an elected fellow of the American Statistical Association, the Society for Clinical Trials, and the American Association for the Advancement of Science. He has published extensively in both statistical and medical journals and has co-authored two influential books, Bayesian Adaptive Methods for Clinical Trials and Model-Assisted Bayesian Designs for Dose Finding and Optimization. Given his deep experience and the interface of Bayesian methodology, clinical practice, and regulatory science, Dr. Lee is exceptionally well positioned to help us interpret the implications of this FDA draft guidance and to discuss how Bayesian approaches may continue to shape the future of clinical trial development. Please join me in warmly welcoming Dr. Jack Lee.
Thank
Jack,
you.
it's your turn.
Thank you. Yeah, thank you so much, Ray Tao, for the kind introduction and also for this invitation for me to present the reflection on the recent FDA guidance. Actually, this is a draft guidance, so still open for comments on the use of Bayesian methodology in clinical trials. So welcome. And so in the next 50 minutes or so, I will try to go through these important points. And also this is a reflection. I will give you some history. And then we can talk about its impact that will be coming. Okay. So again, I'm very happy, just as Rita said, that this is a really timely and important guidance that FDA has issued, right? So let's go through this, and if you have some quick question and just for clarification, you may unmute and just speak. Otherwise, I hope to leave about 10 minutes in the end so we can have some open discussion. Okay, so let's get started. Okay. Whoops, let me see. I don't know why this slide is not advancing. Let me start again. Sorry about that. I tested. Okay, now maybe just a somewhat delay. Okay, so this is the outline of my talk. I will first go through the FDA draft guidance, just to show you what it is, and then talk about statistical inference by frequentist and Bayesian methods. Then we'll talk about Bayesian methods and clinical trials reported in PubMed, followed by Bayesian trials at MD Anderson. Then I will go over the use of Bayesian methods by FDA CDRH, and that's a device branch. And then followed by the use of Bayesian method by CDER and CBER. That's a drug and biologic branch for this FDA. And again, this guidance is issued by the CDER and CBER. Okay, this is recent guidance. Then I will talk about 3 JAMA perspectives that was published in March this year. Then show you some useful resources for the design and implementation of Bayesian trials, and end with some concluding remarks. So many of you probably now are familiar with this draft guidance. It was issued in January this year, again by CBER and CDER. Okay, and this is very important because this guidance has been said here. Okay, that it's actually give guidance for the submitting IND, NDA, and BLAs. Okay, so this is very, very timely and very important. Okay, and let me just change the laser to the laser pointer. So this is the table of contents. So you can see that after introduction and background, and they talk about a situation when Bayesian method have been used. Okay, and mainly in some borrowing information. And as I will also show you that from adult to pediatric trial, for example, and also share information between the subgroups. And the guidance also define the success rate and operating characteristics for the Bayesian method. And then very importantly, talk about the prior distribution. The use of a prior distribution is the bread and butter of Bayesian method, and as you will see in my talk as well. Then they talk about estimates and the missing data, talk about software and computation, and then have some and also how to report Bayesian trial and analysis. Okay, so just a quick review of the statistical inference by frequentist and Bayesian methods. I think everybody is familiar with this, but just to set the stage, we want to contrast the frequentist and Bayesian methods. So basically, frequentist approach assumes data is random and the parameter of interest data is fixed. So we make inference based on the data likelihood of data given the fixed parameter theta. Based on that, then we compute p-values, confidence interval, non-hypothesis significance testing. We know that this frequentist approach was championed by R.A. Fisher in the 1920s, and now it's 2026. So the past 100 years have been dominating the medical field. Bayesian approach, on the other hand, assume data is fixed. Why is it fixed? Because the data you have already observed in clinical trials, so it doesn't change, right? So assume data is fixed, but the unknown parameter theta, for example, response rate, since you don't know, so it's random and has a distribution, and our job is to find that distribution. Okay, so we want to make inference based on the posterior probability of data given data. Based on that, then we can calculate the posterior probability, credible interval, and Bayes factor. Okay. We all know that Bayes theorem was published in 1763, actually 2 years after Reverend Thomas Bates passed away by his friend Richard Price. Okay, and Bayesian 1, 2, 3 is simply prior plus data become posterior. Posterior become new prior, and after observing new data, we can compute the new posterior. So one motto of this Bayesian method is that we learn as we go. So the big question is that can both frequentist and Bayesian frameworks be used for clinical trial? Of course, the answer is yes, right? So the pros of the frequentist framework is that it's well established in medical research and the regulatory setting. And there are many tools available, for example, sample size power calculation and the analysis packages, etc. Okay, and it also seems like this advantage, there's no need to specify the prior distribution. Really? I would say that the frequentist approach actually has an implicit prior and typically is a non-informative prior. Or frequentist approach sometimes, just was implemented by the prior in the statistician head, but not well laid out. The cons of the frequentist approach is more rigid and less flexible when trial conducts deviate from the design. Okay. And also it answers the question of interest indirectly. So for example, okay, p-value is a probability of observing the data as extreme as or more extreme than what you observe if the drug doesn't work. Okay. And again, what we want to find out if whether the drug work or not, but you have to assume the drug doesn't work first, then you calculate the p-value. Okay, Bayesian framework, on the other hand, the pros are you can answer the question of interest directly based on the posterior probability of the quantity of interest. For example, what is the probability that the drug works given the data, right? So the p-value is not the probability of the non-hypothesis true. Okay, you can only answer that question using the Bayesian approach. And it's also more flexible and more adaptive. Prior distribution can put the problem of into the proper context. So we can use the prior distribution to quantify the parameter of interest based on the available data, right? So all the clinical trials that we do, they do not just occur in a vacuum, right? So how do we put the problem into the appropriate context? For example, the response rate for breast cancer and the pancreatic cancer, of course, are different, right? So you can actually formulate that and put it in the prior using the Bayesian approach. And Bayesian approach also can borrow information using the prior or hierarchical based model to increase the trial efficiency. But there are two major sort of cons in the Bayesian approach. One is it's less familiar with research and regulatory settings, so we need more education, and it has less tools that's available and more computational intensive. They often need to get some customized simulation tools, and at the end of my talk, I will show you some available tools to evaluate the trial's operating characteristics. Okay, but caution is this. Bayesian method is not a panacea. You can get the wrong conclusion with inappropriate prior when there's a prior data conflict, and so that need to be known that again, it's not a Bayesian method you can just apply blindly and it will give you the good result. That's just not true. Okay, so just a quick review of Bayesian methods that has been published in PubMed, both in the statistical method and the clinical trial. So I actually use ChatGPT and just let the AI to find in the PubMed what are the statistical papers that publish that has a word Bayes or Bayesian. So it's a very kind of liberal kind of criteria, okay? And as you can see that obviously, you know, it increased over time. And right now it's about 400 papers a year.
Yes, we.
Yes?
Okay.
Can you mute if you're not talking?
Yeah, sure, I'm coming.
Yeah, or Rachel, can you help to mute? Yeah, okay. Okay, so again, you know, this is not surprising, but how about, this is for statistical methods, okay? How about the clinical trials that reported using the word Bayes or Bayesian? Okay, so this is shown in the red curve, red bars. So no, so it definitely also increased over time. Okay, and they now reach about 200 per year. If we put them together, okay, and the figure looks like this. So what have we learned? What we learned is that statistical methods, you know, that definitely become more and more popular, but clinical trial implementation is catching up. Okay, so I think we statisticians definitely help in this trend, and so we got to learn ourselves, getting more familiar about the Bayesian methodology, its use, and its limitations, and how to use it properly, and then implement more in clinical trials. Okay, so next I will talk about Bayesian trials at MD Anderson. Actually, I need to give credits to Don Berry, who came to MD Anderson to establish the biostatistics department in 1999. And since then, the MD Anderson has faculties and staff and trainees have actually on board on learning and using the Bayesian method, and we have been leading in terms of the develop methods and then implement them in clinical trials. So in 2009 in Clinical Trial Journal, we published this review of the Bayesian clinical trial used at the MD Anderson Cancer Center. And Ted Gonen, good friend in the Memorial Sloan Kettering, has an editorial, and basically he says, Bayesian clinical trials, no more excuse. Okay. Actually, not implemented, not using it. He says that while there are certainly some at the other centers using the Bayesian method in clinical trials, the bulk of applied Bayesian clinical trial design in this country is largely confined to a single zip code. Why is this the case? Okay, and that he very properly point out that there are three barriers: okay, prior, software, and motivation. And we got to do better than that. And this was 2009. Then in 2019, 10 years later, we published a follow-up paper to summarize the Bayesian clinical trial at MD Anderson. Okay. And this is the result. Okay. On the left panel shows the first 5 years, and then the right panel shows the later 5 years. As you can see, these are the IIT trials at MD Anderson, investigator-initiated trials, and then the blue areas are the Bayesian trial, okay, and orange areas are non-Bayesian trial. As you can see, that the Bayesian trial, okay, is about over 50 trials, IIT trials in recent years. So if we put it into the table, and the top panel shows the earlier 5 years, the bottom panel shows the later 5 years, and we can see that in the earlier 5 years, there are about 20% of the trial conducted at MD Anderson use some of the Bayesian method, not necessarily designed as a Bayesian trial to begin with, but use some Bayesian method in analysis in reporting as well. Okay, and in the later 5 years, then the percentage of Bayesian trial increased from 20% to 28%. If we stratify by MD Anderson only or multi-center trial and by pharmaceutical sponsor or other trial, as you can see that if it's the MD Anderson only trial, actually over 50% of the trial has some Bayesian component. Right? And for multi-center trial, it's only about 14%. If we stratify the trial by phase, this is what we see. Right? So mainly the phase 1, phase 2, and phase 1/2 trial has about 40% has some Bayesian component. But here, as you can see, phase 3 trial is only 1%. Okay? And here, the phase 2/3 trial only 13%. Here, even in the later 5 years. So it's still mainly used in earlier phase trial compared to the later phase trials. So what are the components that has the trial used in the Bayesian elements? Okay, so we can see that the most common two components are for efficacy monitoring and toxicity monitoring. And also we have some trial using adaptive randomization and also for dose finding. Okay, some trial using predictive probability, hierarchical model, etc. Okay, next I will talk about the use of the Bayesian method by FDA CDRH. Okay, and this is really credit to the a good friend, the late Dr. Greg Campbell. He has been led this effort in the device branch of the FDA. As you can see that even in 2010, okay, so that's 16 years ago, this guidance for the use of Bayesian statistics in medical device was published. Okay? So that they have, actually led the effort and really implement the Bayesian design and analysis in the device. So this is the table of contents of that guidance. Okay, and you can see that they talk about Bayesian statistics, how to plan a Bayesian clinical trial, okay, how to analyze the Bayesian clinical trial, etc. So this is a document you can find in FDA. Basically, they list that there are 44 devices that approve employ Bayesian statistics. Okay, this is up to 2022. Again, the common use are using the posterior probability and the predictive probability hierarchical model, borrow historical information. And I will make my slide available. And in this slide, it shows all 44 devices that are approved by CDRH, by FDA CDRH, that you apply Bayesian method and what did they use, okay? And as you can see here, you have predictive probability, you have hierarchical model, you have some Bayesian interim analysis, etc. Okay, and there's the link for the reference. So these two tables simply show us that the device, what are type of the device, by what company, what dates of approval, what Bayesian method used, and the link. So if people are interested, this will be very useful resources. Okay. However, the drug side and biologic side, the CDER and CBER, in terms of applying the Bayesian method, kind of lag behind. Okay. So this table shows that there are 4 different drugs recently approved by FDA, and what are the Bayesian method they use. Okay, using dynamic borrowing, beta binomial model, Bayesian meta-analysis using these robust MAP prior or Bayesian hierarchical model. And let me just give you 2 examples. So the first is the information borrowing of pediatric trial for lupus. And pediatric lupus is a rare disease, so typically, there are not enough patients in the trial that can reach the high statistical power. However, there are data in adult trials. So can we use this adult data and borrow that information to pediatric? And so this for this drug, this trial, they use a robust meta-analytic prior to borrow information. So what they found is that the prior weight of 50% or larger on the adult data will provide the 90% credible interval, exclude the odds ratio of 1, which says no difference between the experimental drug and the standard of care, or the supportive evidence. Okay, and so they calculate the effective sample size and correspond to about 83.4% borrowing of information, and this drug was approved by FDA in 2019 in the pediatric patient settings. So this table shows the information borrowing from adult to pediatric trial. So there are actually 2 adult trials, okay, as you can see here on the left. And then on the right is the result of the pediatric trial. And you can see the response rate, okay, for each trial. And then if we put, and you can calculate the odds ratio accordingly. So if we put into the graph, this is what you will see, okay? So the x-axis is the prior weight, the y-axis is the odds ratio, okay, and 1 is a dashed line, shows no difference. So if we only use a pediatric patient that there's a 0 weight on the prior borrowing, as you can see that this credible interval that definitely covers 0, right? Even though the treatment shows that the odds ratio here, it's a beneficial as a point estimate. And on the right here shows the credible interval for the 2 adult trial. Okay, so if we borrow this information, and you can find that the prior weight of 55% or higher by weighting the adult information, then the credible interval will exclude 1. Okay, that shows the drugs are effective. So the second trial is the trial on C. diff infection. Okay, try to prevent this infection using the Bayesian hierarchical model. So there are 2 trials. One is the phase 3 trial, and one is the phase 2b trial in a similar setting. So, and the model-based estimate, you can see that the success rate, okay, of the 2 trial, the new drug is 70.6% versus 55.7%. So there's an estimated treatment effect of 13.1, and the posterior probability of superiority is over 0.98. Okay? So this drug was approved by FDA on November 30th, 2022. And if you look at the data here, okay, and this basically shows the success rate, okay, and based on the ITT analysis, and probably better to be shown in this graph. Okay. On the left, it shows the density of the placebo and the drug, the new drug. Okay, and then on the right, it shows the posterior probability that treatment that is more efficacious than the control, and with the posterior probability calculated in the shaded area. Okay, so now I will move to the JAMA perspectives. So after publishing of the FDA draft guidance, and JAMA invites 3 groups to write perspectives, and I also, along with a good friend, Frank Harrell, Lisa Lavange, she used to be at FDA, and actually before that and after that was at the UNC, and then Sir David Speaker-Halter. Okay, so we published one of them, and I will mainly talk about our perspective, but then I will also cover the other 2 perspectives. Okay, so what did we say? Oh, by the way, if people are interested, you can find it in JAMA, and So what we say first is that this is an exciting and welcome guidance for the therapeutic trial design and analysis that we expect to contribute significantly to the rigor, efficiency, and flexibility of clinical trial. As I've shown you, look at the Bayesian method have been applied over 30 years in clinical trials. Okay, so it's not something new, right? Actually, Spiegelhalter has published these two landmark piece of papers. One is in 1994, the other is in year 2000. And basically lay out the Bayesian approach we used in the randomized clinical trials. And that's when, 1994, it's actually 32 years ago, okay? So it's been long, and really a challenging road that to apply this in later phase trial for FDA's approval, particularly in the drug and biologic. But we are happy that our FDA colleagues also now catching up and then provide this draft guidance. Okay, so this new draft guidance lay out fundamental concepts and approaches to apply Bayesian methods in therapeutics that dispels the misconception that FDA opposes the use of the Bayesian method in drug development. So very important. And one central question in drug development is that does a drug work? As I've shown you earlier, frequencies provide the indirect answer to the question of interest, but Bayesian provides the direct answer. Okay. Its Bayesian method is also more intuitive and easier to understand in the simple and complex application. And actually, I found that it's also easier to explain Bayesian result to clinicians. If you try to explain what's the p-value, what's the confidence interval, in the right way, actually, it may not be easy. Also, it substitute, the Bayesian method that actually substitute a single transparent decision, the choice of the prior, for a host of other the more complex settings like statistical versus clinical significance, multiplicity adjustment, non-inferiority test, and sequential adaptive design, use external information. All this, can be properly put into the prior and then actually use it accordingly. But it's very hard to do this in the frequencies analysis. Also, in this JAMA perspective we wrote, the title is actually Embracing Bayesian Method in Clinical Trials. And Bayesian model has its own operating characteristics. So you can actually do simulation to find the feature of the Bayesian method instead of just focus on type 1 error rate and power. Right? And sponsor may choose a Bayesian operating characteristics if a specific prior is well justified. So the well justified is important, okay? And this need to be communicated with FDA and agree upon. Then you do simulation to find out the operating characteristics. Yeah. And you can also vary the prior distribution by sensitivity analysis and to reach the high decision reliability. Okay. The guidance illustrates the problem-solving capabilities of the Bayes model. Okay, first is borrowing information. If you have a high-quality real-world data, okay, and then the Bayesian method allow you to join modeling and borrow information to increase the efficiency of your trial. And Bayesian hierarchical model can be applied for dynamic borrowing information among the subgroup, like in basket trial, to sharpen the estimation of the treatment efficacy in each subgroup. And also you can actually get a collective kind of the overall estimation of the treatment effect for the whole trial. High quality perspective comparative data are applicable. Again, as I showed you that how to borrow adult trial information to a pediatric setting of phase 2 to phase 3. trials. Also, another unique features of the Bayesian method is that you can continuously learn about the parameter of interest and do the interim analysis accordingly. Again, as I told you earlier, the one motto of the Bayesian method is that we learn as we go, right? So applying Bayes' theorem is simply synthesizing the information contained in the prior and the data to form the posterior, and you just keep doing it. So Bayesian model is innately adaptive, and no special allowance needed to be made in analyzing this data. Okay, as adaptation goes as the trial move along. Continuous learning can guide the interim toxicity and efficacy monitoring, but to preserve the study integrity, the key adaptive design features and parameters need to be pre-specified, and this is very, very important. You cannot, after you observe the data, then you try to find the prior, to make it look good. That's not allowed, right? And or you can whatever interim monitoring scheme, you need to be pre-specified so you can, in the trial conduct, you can follow accordingly. Also, the use of skeptical, optimistic, and non-informative prior distribution are allowed. Skeptical prior can be used for non-curative therapies in the absence of strong external data, and pre-specify of the prior assumption need to be clear, and actually